So if you recall, we started out talking about our resting membrane potential. Which is at a minus 70 millivolts. Some neurons it's around a minus 90 or a minus 55. And what we're talking about is that right at the plasma membrane, the outside of the cell has more positive charges than the inside of the cell, or at least the inside of the cell has fewer positive charges, and so we see this relationship. And that's what the negative means. So the inside the cell is negative in relationship to outside the cell. The minus 70 comes from the fact that if we were to open up the membrane and allow ions to flow into or out of the membrane, they would flow into the membrane. They would be positive charges. They would flow into the membrane, and a sufficient number of them would flow into the membrane to create a current of 70 millivolts. Okay? That current of 70 millivolts is what would be needed to bring, to change this appearance and bring it to no difference in charge across the membrane. That's what the 70 millivolt number means. Is that's how many ions measured as an electrical current would be required into the cell to bring us to a zero resting membrane potential. All right? Now, when we talked about the action potential, what we saw was a spike that occurred above 0. Typically this spike is at around plus 30. Right? What that means is that with the opening of the voltage-gated sodium channels, we have the inside of the cell is more positive than the outside of the cell. Okay, so this is resting membrane potential. This is action potential, such that if we were to allow ions to flow through our channel, they would flow in the opposite direction. We have more positive inside than outside. It's right at the membrane. So there would be an outflow of positive charges such that it would take a current of 30 millivolts, again, to bring this back down to 0. Not to bring it back down to minus 70, just to bring it back to 0. Okay? That's what the plus 30 means when we're at the peak of our action potential. All right. So just explaining again those numbers. Now, we talked about the resting membrane potential. We have our threshold, which is usually around a minus 55. Again, that can vary, but it's often around a minus 55. It's going to vary with each neuron. So let's say that on our neuron, and I'm going to, may I erase this over here? You guys have that already? I'm just going to draw this line across the board here, so I decided I would put my next pictures over here. Okay? Let's say we have our cell body. We have a presynaptic neuron releasing neurotransmitters. Here is our postsynaptic dendrite or dendritic spine. And we'll make this a ligand-gated channel. And this is going to allow positive ions into the cell when our neurotransmitter binds to the receptor. So the neuron fires and one or just a few channels are open. This is allowing positive ions inside the cell. So what's going to happen to our resting membrane potential? It's going to depolarize. So that's a pretty small current. So we're going to see just a small, maybe even smaller than that, upward deflection. And then it returns to a resting membrane potential as those ions, maybe positive potassium ions leave, or these are removed via our sodium potassium pump. And so this is a graded potential. Because it did not bring the change in the resting membrane to our threshold, it is considered to be a subthreshold potential. Now, let's have this neuron fire repeatedly. Okay? So it's going to fire multiple times. Obviously, it can't fire at the same time because it's the same neuron. So if I'm going to hit this table really fast, I can't hit it 10 times at the same time. That's just impossible. Okay? I have to hit it really fast 10 times. So I'm going to see an effect of each of those changes. So if I cause this to fire, I'll just make it 3 times, times 3. I will see a change. I'll see each of those effects in that occurrence, okay? Because I'm causing that channel to open more rapidly. And so I have, what do we call this when we have the same neuron firing multiple times?
Temporal.
Exactly right. This is temporal summation. So with temporal summation, I can see an effect of each of those very rapid, almost on top of each other, firing of the neuron. Now let's say I have another neuron over here. And it also opens a channel that allows positive ions into the cell. And so if it fires, let's say this is neuron A, and this is neuron B, right? So this result here would be neuron A1 plus A2 plus A3, firing 3 times. Okay. So now let's have neuron A and B fire. They can both fire at the same time, right? So if each one fired by itself, let's say that's neuron A, and let's say that this is neuron B. If they each fired by themselves, they can fire at the same time. And so if we add both of their effects together, we add them. So this is another form of summation. Spatial summation. So this would be A plus B. Okay. Now A plus B may be enough, I think it is in the way I drew it here, may be enough to reach threshold. Oh, no, not quite. Good, I didn't want it to. Okay, so it's still subthreshold. All right, we haven't reached the minimum current required to cause our voltage-gated channels to open. It looks like my resting membrane potential is slightly going downhill. Maybe otherwise it would have been enough to reach threshold. Okay? Now let's add a third neuron. When this neuron opens a channel, chloride goes in. Okay, what's that going to do to my resting membrane potential? It's going to cause it to hyperpolarize, right? It's going to drop it, make it increase the difference between resting and my membrane potential, threshold and my membrane potential. So each of these events results in depolarization, which makes the cell more excitatory. If I were to have another neuron fire at this time, I actually might go over a threshold. Okay? So each of these is an EPSP, whereas this one is an IPSP. Is it still a graded potential?
Yes.
Yes. Okay. Now let's do a spatial summation of A and B and C. Here is my summation of A and B. All right. Now let's do A and B and C. I'll just draw it in black. So A plus B plus C as a spatial summation. It's going to result in this. Still excitatory, but not as much because of I had to fill in this with a bunch of positive ions, so my threshold did not go, my membrane potential change, my depolarization was not quite as high. Does that make sense? Okay. Would I ever get something like this with a temporal summation?
No.
No, because one neuron is not going to have both an excitatory and an inhibitory effect. Okay? It's binding to the same ion, the same channel. It's going to just multiply that effect. It's not going to counter-effect itself. So if you're going to have the sum of a positive and a negative, that has to be a spatial summation. Does that make sense? Yes.
Can you have spatial and temporal at the same time, like three neurons firing?
Yeah, so this neuron, if this neuron had a temporal summation, then I might even get a flat or end up with an IPSP. This one could be temporal added to the spatial of A and B. So it would be spatial A, B, and C. They'd all be added together, but C could also be temporal and end up doing that. Okay, so yes, that could happen. Yes?
This is different than the pump, though, right?
Right.
Okay.
This is, the pump doesn't have to have, we don't even have to be in a neuron. The pump would be how we would recover from this.
Okay.
So it's there,
But from
but
like
it's
the
not
action.
a part of the specific event.
Okay, but from like when we do the action potential and it goes down and it hyperpolarizes, this is different.
This is different.
Okay.
And part of that is also the fact that our potassium channels are still open. The pump is how we're getting, why we're not staying here, why we're getting back to this. Okay, along with the leak channels. Okay, so all of these events that I've shown you are still graded potentials. Okay, very short. They are not self-perpetuating. They can be of different degrees of depolarization, different degrees of hyperpolarization. Okay, any questions on these? All right, let's say now that we have, whether it's temporal summation or spatial summation, let's say that finally one of these reaches threshold. Okay, so with your permission, I'm going to erase this board right here, and we'll start again with action potential. Okay, is there a question, please?
Oh, no.
Okay. It's clear as mud, right?
Of course. It's
It's like 1 plus 1 is 2, 1 plus a minus 1 is 0. Okay, 1 plus 3, 2 is 3. 1 plus 2 plus minus 1 is 2. The same thing as adding positive and negative numbers. We're just adding positive and negative charges. Okay, anybody else? All right. So let's have a strong stimulus, and when a stimulus is shown as an arrow like this, the length of the arrow is, this would be a stronger stimulus. Okay, yes?
IPSP is
Inhibitory postsynaptic potential. Okay, so again, that's what's happening. This is the postsynaptic neuron. So this would be a EPSP, because we're adding positive charges. This would be an EPSP, because we're adding positive charges. This would be an IPSP, because we're adding negative charges, or we could lose, we could lose positive. Okay? So this could either be chloride, or if we were losing, we could do another IPSP by losing potassium ions. Either of those would cause an IPSP. All right, so we have a stimulus. This first stimulus resulted in a subthreshold EPSP. Subthreshold. Okay? Did not reach our membrane potential. So no action potential was created. So let's add a stronger stimulus. And actually this should be at the beginning. So we add a stronger stimulus. Now we reach threshold. Okay? And that would result in a threshold potential, a current strong enough to open our voltage-gated channels. Before we get to the action potential, however, I want to illustrate something that we talked about on Friday. Boy, keep drawing, going downhill here. And that was, remember what happened if we added potassium ions to the outside of the cell? We increased extracellular potassium ions. What effect would that have on our leak channels for potassium? Not the leak channels, but the intracellular potassium ion concentration.
It would go down.
It would go
It
up.
would go up, which makes
Okay.
it less.
So this would in effect block potassium ion diffusion. Out of the cell. Therefore, it would increase intracellular potassium ion concentration. Okay? Yes?
So what would that do to hyperpolarization?
It would make it less likely to happen, because this is what happens. So if we do that, which way is my resting membrane potential going to move? Going to get closer to threshold. So here is my new resting membrane potential. Now, here's my subthreshold stimulus. It now ends up being of sufficient strength to open my voltage-gated channels. It may not. If it was weaker, it might not be. Okay, but at least it's more likely to, because my resting membrane potential moved up. Does that make sense? Is what we talked about on Friday with that table in the lecture notes. So if I add potassium ions outside the cell, this would be the effect of an increase in extracellular potassium ions. It raises, or not, it brings my resting membrane potential closer to threshold. It makes the cell more excitable. So a subthreshold stimulus is more likely to result in an action potential. Okay, now let's look at what happens if we decrease extracellular potassium. What's going to happen?
It's going to bring your resting potential below.
It's going to increase the loss of potassium, and it's going to have the effect of lowering my resting membrane potential such that even a threshold stimulus might not be high enough to reach my normal threshold potential, and nothing with the cell would not fire. Okay. So this would be a decrease, not that here, but a decrease in extracellular potassium concentration. Okay. So my cell is less likely to fire. Now the same thing is happening here by doing an IPSP. The difference between these two is this is affecting the entire resting membrane potential for a longer period of time. This is transient. Okay? It has to be added, it has to be occurring at the same time as I'm doing one of these to have an inhibitory effect. Because this is going to result in this, and it's not going to last very long. Right. So if this doesn't fire at the same time, this will be gone by the time that one fires later and won't see an effect. But changing my extracellular amounts of concentration of potassium would be a longer period. I have a potassium IV or something like that, or a drug that takes potassium out of my system. That would have a longer effect. Okay. Erase this and move on finally to our action potential. So if you guys want pictures, go ahead. Get out of the picture. Okay? All right. Now, finally, so when we have our resting membrane potential, what is the position of our sodium and potassium voltage-gated channels?
Wait, what was the question?
Yeah, what'd you ask?
Okay, resting membrane potential, that's the straight line.
Yep.
A voltage-gated sodium channel is going to be open or closed.
Domino.
Close?
It's going to be closed. And so it's going to look like this. With the wrong colored caps on here.
Really.
Okay, it's closed, but
Open.
the inactivation gate is open. And my potassium, voltage-gated potassium channel
This is closed.
is also closed. Okay. During the resting membrane potential. Then I have a stimulus. And I create a graded potential which reaches my axon and is of threshold strength. So this is a resting membrane potential. This is the graded potential. This is the threshold potential. So does anything change for the graded potential? As far as my voltage gated channels are concerned?
No.
No. Okay. But as soon as I reach threshold, what that means is my voltage gated sodium channel is going to open. And my voltage gated potassium channel isn't yet open. Oh, I'm drawing Stonehenge rock here. The same image applies once my threshold is open. I rapidly depolarize. So this is my voltage gated sodium channels are open. And I'm depolarizing. So starting with threshold and continuing through depolarization, my voltage gated channels are in this position. The same voltage opens my potassium gated channels, but it takes them longer. So my voltage gated sodium open and closed, and then I reach the peak of my action potential. And the reason that's the peak is because voltage gated channels close. No more positive ions are coming in. All right? So we get to the peak, and my voltage gated channels close. This time, it's the internal gate that closes. So that's the reason I have a peak. And then I begin repolarization. Okay? And repolarization is due to the opening of voltage-gated potassium channels. So my voltage-gated sodium channels have closed, and now my voltage-gated potassium channels open. That allows potassium to leave, and so I am losing positive ions, and the inside of my cell is becoming less positive, and so this is repolarization.
Is there a reason why it's the potassium that goes out and not the sodium, since the sodium came in such an abundance in the first place?
Because of the specificity of the channel.
Okay, so just because,
Yeah.
pretty much.
Some sodium could go out, but most of, like 1,000 to 1 or 10,000 to 1, it's going to be potassium
Okay.
because of the interaction of with the amino acids forming that channel. All right, notice we went below 0. So that's because, not below 0, but below our resting membrane potential. That's because our sodium-potassium channels are still open, and we haven't quite recovered because of our sodium-potassium pumps. And so we have hyperpolarization. So at this particular voltage, in hyperpolarization, it closes all the gates of our sodium, and our potassium are still open. And then finally, and again, this is due to current. Remember, these are voltage gated, so the reason each of these are opening and closing is because of the particular voltage. So as we return to our resting membrane potential, we get to this, okay? The potassium voltage gated channel will close, and we open the lower portion of our voltage gated sodium. So when we return to resting membrane potential, we come back to this appearance. That was almost all of Friday's lecture in 30 minutes. Two more things I want to talk about. One major thing. What is that?
Refractory period.
Refractory period. I don't want to draw it on that diagram because I already have too much stuff on it already. So, this time point is the refractory period. Okay? Starting with the threshold, because during the graded potential, we're not in a refractory period. We haven't opened our voltage gated channels yet. So the refractory period starts with the opening of the voltage gated sodium channel. Okay? When we get to hyperpolarization, we change. So during this time, this is the absolute. Refractory period. Meaning that no matter how strong our stimulus is, we could make the inside of the cell a plus 60, and we're not going to open, we're not going to affect for the first part because our sodium channels are already open, okay? And for the second part, it takes a stronger current to open them when they're in this position. So this portion, so which of these events apply to absolute refractory period? This one and this one would be absolute refractory. Okay. And then this period, and I have them looking like they're the same time, they really aren't. That will open with a strong enough current, and that the sodium gated and the potassium gated channels are in that position. Now, in a couple of weeks, we'll be extending this information to the changes that occur in a skeletal muscle membrane, and it's going to look exactly the same. We have voltage-gated channels and ligand-gated channels and all of that in a skeletal muscle cell. In a skeletal muscle cell, here's the length of time for an action potential. Here's the length of time for a skeletal muscle contraction. So they're almost the same. Okay. In cardiac muscle, so this is skeletal muscle. In skeletal muscle, again, it's not a neuron, it's the muscle, but the same events occur. This lasts about 1 to 2 milliseconds, 2 to 3. This lasts 200. This 300 milliseconds. It lasts almost as long as a little bit longer than the refractory period. So that with a skeletal muscle cell, I can actually trigger a skeletal muscle cell to contract before it's completely recovered. We wouldn't want to do that with the heart. Because a skeletal muscle, you've probably heard of tetanic contractions technique. Botulism causes tetany in muscle cells where they go into a spasm and they won't relax. That would result in death if your heart went to a spasm. Because when it's contracting, blood flow isn't occurring. Blood is not returning to the heart, and it's not pumping any more blood out. And so we have to prevent the cardiac muscle from going into tetany, and we do that by prolonging that refractory period. Notice how more gradual this slope is? Because while potassium is going out, calcium is coming in. So we're losing positive ions, but it's not a sharp drop because we're also gaining positive ions. Okay? The reason we get this sharp drop here is because our positive channel for sodium closes. If it was to continue to stay open after the voltage-gated channel opened, we would get a little more gradual slope. The reason we see that sharp drop-off is because the positive channel for the gain of positive ions has closed. At the same time, the loss of positive ions is open. And that gives us our sharp view. So just in review, that was almost a 45-minute lecture. Why don't you guys take a short break? All right. Just to clear your brains. And then
What?
I was wondering for somebody's reaction.
700 a day.
I think I was too much of a shock. Just 700? Like, I knew this was a thick thing.
There is a lot of information on these pages that I'm not going to actually say or necessarily hold you responsible for on the comprehensive, but that you will need to answer the take-home exam. Okay? You will need more than is on these pages to answer the take-home exam. So this is more as kind of expanding tables that are in your textbooks, and I'm giving you some general guiding principles, kind of along the effect of the more often you hear the words, the less afraid you will be of them. Notice I didn't say understand them, but be less afraid. Okay? It's one of the reasons why I have such a hard time reading research papers or articles on genetics, because everybody loves to use acronyms and initials. And it's the CGI gene or the OncoK gene. And it's like, well, if you told me that CGI stood for, class, grades, inhibited or something like that, then every time I saw CGI, I would know what I was talking about. But it's just a block to my understanding of what's going on because I don't know what the abbreviation stands for, and therefore I can't understand anything that you're saying about it. So in that line, every time you see 5-HRT or SSRI, you don't know what it means, then anything that's said about that afterwards is going to be difficult for you to understand. Okay? So we're going over this to give a baseline of information, but not to the level that you have to diagram it on your own or repeat steps of activity in that essence. I'm trying to get you more comfortable, more familiar with the processes, so that when you read about them for the take-home exam or when we get into them for lecture next year, it won't be as frightening. It won't be as unfamiliar. You won't be as likely to say, I don't know what this is talking about, and move on. Okay? So that's kind of the purpose for today's lecture. The first few slides are the very first lecture of second semester. So if you do take me for second semester, the rest of the class that hasn't had me will be completely lost, and you'll be going, oh, I remember that. You may not know it, but you won't be that, oh dear, this semester is starting off horribly. I have no idea what she's talking about, and this is only the first day of class kind of thing. In the first semester, second semester, we'll be looking at these for hormones. What do hormones do? How do hormones change cell behavior? Today we're looking at them, instead of as hormones, as neurotransmitters. All right, instead of getting into the blood and traveling all over the body to find a receptor to bind to, they only have to cross a very short synaptic difference space between two neurons. So obviously, as a hormone, as a neurotransmitter, it is much more quickly acted on and much more quickly removed from your body. For instance, we're going to be talking about sympathetic nervous system definitely by Tuesday, maybe as early as Friday. And norepinephrine is a neurotransmitter for our sympathetic nervous system, the flight or fight. We also happen to have an endocrine organ, the adrenal gland, which makes norepinephrine. Okay, the difference between the two, as a neurotransmitter released by sympathetic fibers in your heart, it has a very short distance to travel to get from the neuron to the muscle. And it's quickly broken down by being taken back up into the neuron. As a hormone, it gets into the blood. It takes longer to have an effect. It takes longer to remove. So again, let's say that you're driving down the freeway, and you're going 75 miles an hour, which the rest of the traffic is doing, and a CHP car gets on the freeway and comes on right behind you. And your first response is, okay, I know everybody's going 75 miles an hour, but he's right behind me. And then he turns his lights on. And you're like, okay, it was my luck of the draw. I'm the one he picked out today. And what happens, your hands get dry, sweaty rather, your mouth gets dry, your heart rate starts pounding. And you're like, oh my goodness, I have my Christmas money to spend, I don't need a ticket. And then he pulls around you and goes on down the road because he's been called to an accident. As a neurotransmitter, that norepinephrine should be gone in milliseconds. You're still 5 miles down the road, 10 miles down the road, your heart is still beating rapidly. All right, that's because the norepinephrine that was released from your adrenal gland is still in the blood. It has a much longer half-time. It takes a lot longer for it to be removed via the liver, via your urinary system. So that's kind of the difference between hormones and neurotransmitters. Okay, the hormones are in the blood. They bind to the same they bind to the same receptor, but it takes less time for that to occur, less time for it to be broken down. Now, not all of our neurotransmitters are hormones. Okay, I'm just illustrating. We'll be seeing these slides next semester when we talk about hormones. But the hormones have gotten to those receptors via the blood. The neurotransmitters are getting to those receptors across the synaptic space. Okay? Yeah. Not all hormones, but norepinephrine that comes from a neuron in the nervous system and norepinephrine that comes from the adrenal gland are the same, because those cells in the adrenal gland, actually embryology, we were neurons. They're just no longer connected to the other components. So yeah, the chemical component makeup is the same. All right. So back when we were talking about ligand-gated channels, and the rest of today's conversation is concerning ligand-gated channels, with one exception, which is unusual. Now, when we first talked about ligand-gated or ligand-gated channels, I'm just going to choose ligand. Okay, I'm tired of saying it both ways every time I say it. So ligand-gated channels, chemically gated channels, and no longer going to say chemically again, it's going to be ligand-gated channels. Ligand means ligand and chemical, okay? These are, we talked about the direct pathway. Remember when I talked about direct? What was the key to the direct?
It's directly open.
Okay, the channel itself was the receptor, or the receptor is the channel. Our new term for this is ionotropic. Okay, so ionotropic receptors when the neurotransmitter binds, it opens and the ion passes through. Okay. This is very quick, doesn't take a lot of time, for this event to occur. It's quick to happen, quick to stop happening. The indirect pathway, we demonstrated that the receptor for the neurotransmitter was not an ion channel, was not a channel at all, but involved what we're going to be discussing next as a G protein pathway. So in this one, downstream events ended up resulting in the opening or the closing of a channel. So several steps later, we see this event. Now, as a hormone, it may, that several steps later, may not result in a channel opening or closing. It may just result in enzymes being activated that do other things inside the cell, right? So for the hormones, it may be some different result. What is different, what makes this different, whether it's closing or opening or closing, is due to specific enzymes that are present in the cell. Are activated or inactivated. So with an indirect pathway, it may be excitatory or it may be inhibitory depending on what that cell, the cell that has the receptor, is. Because the neurotransmitter is the same. So this is known as, a new term for this is metabotropic. Because of these several steps, it takes longer, okay? It may take a few hours before we see this effect. It may take minutes or, seconds rather than milliseconds or microseconds. So this is our metabotropic pathway. And we're going to go into much more detail than this.
So we're dropping names direct and indirect to
Ionotropic,
ionotropic.
ionotropic, and metabotropic.
Okay.
Now, not only, that seems to be a detrimental result, is that it takes longer, but a positive effect is that we amplify it. Here, one neurotransmitter opens one channel. In this case, one neurotransmitter will activate one G protein, but that can activate 100 molecules in a cell that each open 100 channels. So we can amplify the effect, all right? And it may be good or bad, depending on the disease. If there's a disease involved, that it takes longer. So you'll notice down here we talk about a G protein. The G protein term is used for guanine, and it utilizes a GTP molecule. Remember, ATP is how we store energy. GTP is very similar. It's an energy storing molecule. It's guanine triphosphate instead of adenine triphosphate. But that's where the G protein comes from. So this is actually a slide from next semester. And there are three mechanisms, three pathways, three methods by which a G protein can change the inside of a cell's behavior. These have to do, the slides are showing a hormone, okay? Most of them are hormones, but they apply to our neurotransmitters as well. May I erase the board? Okay. So they all have a common step. And that is activation of the G protein.
Oh yeah, I have that.
So this is our neurotransmitter receptor. It could be a acetylcholine receptor or a norepinephrine receptor or a glutamate receptor. and it's going to bind the neurotransmitter. When it does so, the receptor changes shape as a result of the changing interactions, bonds that are formed between the neurotransmitter and the receptor. And attached to the receptor within the postsynaptic neuron is our G protein. It has an alpha segment, a beta segment, and a gamma segment. Typically we deal just with the alpha, occasionally with the beta, right? But most commonly the alpha, when it's an excitatory component. Beta and gamma tend to be an inhibitory effect. All right? They stop activating an enzyme instead of acting an enzyme. Here the excitatory inhibitory doesn't have anything to do with positive or negative charges. It has to do with turning on or turning off events inside the cell. So when this binds, the G protein separates from the plasma membrane, and the alpha segment is activated by GTP. And now it has a different shape and it can do different things. So that's the common step, okay, is in the activation of the alpha subunit with the GTP molecule. Here are three different things in an active G protein mechanism by which the active G protein can act. Two of them involve the formation of a second messenger. So, to illustrate what I mean by a second messenger, notice the neurotransmitter doesn't go inside the cell. It stays bound to its receptor. So let's say that I live in a tiny little house next to a huge estate, okay, that's made up of 10 different buildings. Each person in the family has their own building. And they like to leave security lights on all night long. And I can't sleep when those security lights are on. So a direct act would be for me to walk over, open up the door, go inside the house, and go to every building and turn off the security lights. That would be a direct response. That would be me entering the house, going into the cell. Well, that would be breaking and entering, and I would only do that one time and then have consequences, okay? So instead, I'm going to be a little bit more polite, and I'm going to go knock on the door and say, I can't sleep, would you mind turning off your security lights? That would be the neurotransmitter binding to its receptor. I don't go inside the house, okay? But when I knock on the door, somebody comes to the door and opens it. There's an effect. And I say, would you please go turn off your lights? And so the butler or the maid or whatever goes to the owner of the house and the 5 kids and says, your neighbor would like you to turn off your lights. So all 6 people go to their bedroom houses, if you will, and turn off the outdoor lights. So just me knocking on the door activated 6 people inside that house to go do something. The person who I talked to, when I said, would you please tell them I can't sleep, would be the second messenger. He would be the person, he or she would be the person that went inside the house and got those other people to do something. I didn't have to go inside the house. Okay? So the second messenger would be the person doing that work for me, giving the message. So let's see how that works with these 3 different mechanisms. We have activating of cyclic AMP, which is our second messenger, calcium channels, which doesn't require one, and then the IP3DAG, which is a combination of those first 2. So this is the cyclic AMP. Yes?
Is the guanine bound to like all of the?
The guanine is the GTP.
No, what I'm saying is the guanine is it bound the GDP rather is that bound to the alpha, the beta, and the
Yeah. So what this was GDP. So in other words, that's not shown on here, but it starts out as the energy is used, and GDP is formed, and then GTP comes and knocks off the GDP, and it's bound as GTP.
So what I'm asking is that does that only occur in the alpha?
Yes. These have other effects, but yeah, the GTP is binding to the alpha. Yeah. All right, so that was the common component. Now let's look at what happens with cyclic AMP activation. This is our second messenger. So I'm explaining this at the level for next semester. Some of the steps, and this information I'm going through right now is not something you need to know for the comprehensive exam. Because there's always this conflict about, okay, well this is a take-home test, but the comprehensive isn't take-home. How much of this do I have to know? All right, so I will keep explaining levels of required memorization. Now, cyclic AMP is a form of ATP. When the alpha component is activated, it bumps into an enzyme inside the cell membrane known as adenylate cyclase. Anytime you see ASE on the end of a word, it's an enzyme. Now, not every enzyme ends in ASE, okay? It ends in IN, but so do hormones. But anytime it does end in ASE, it's going to be an enzyme. Enzymes work by increasing the rate of the reaction. They kind of start the reaction going so it's more likely to happen. So now we've activated our adenylate our adenylate cyclase, and in that active form, it will bind ATP and convert it to cyclic AMP. That's our second messenger. Okay, that's the person answering the door. Cyclic AMP then will interact with a variety. We have molecules inside the cell known as protein kinases. Notice these are another enzyme, but I'm amplifying my effect. One cyclic AMP molecule can activate multiple protein kinases. And what protein kinases do is they phosphorylate. They add phosphates. Sometimes they remove them to other enzymes or proteins to activate them. So again, sometimes they can deactivate them. So what we're going to see in our neurotransmitters is that the activation of these protein kinases are going to end up causing a channel that is closed to become open, or vice versa. All right, so depending on the postsynaptic neuron, we'll either open or we'll close channels. We have to go through this entire event, all of these different steps, to get our ion channel to open. The receptor doesn't have any space in it for the ion to pass through. So yes, it is time consuming, but we have a whole lot more ion channels that can open in response to one neurotransmitter binding. There's an amplification that occurs, and it usually lasts longer. Anytime we have more steps like this, it also gives us more places to control the reaction. And one way of controlling the reaction, of course, is removing the neurotransmitter. So we'll talk about that in a bit. Another way of controlling the reaction, because eventually we want this to stop. We don't ever want our synapses to be always and always and always active. So another way of controlling this is to remove the cyclic AMP. The enzyme that does that is known as phosphodiesterase. And it results in inactivating cyclic AMP. The reason I mention this here is we're going to talk about it next week when we talk about how the retina of the eye works. We're going to use this pathway. So that is the first of the 3 different ways that G proteins work. Is the cyclic AMP. It's probably the most common of the ones that we see with our metabotropic neurotransmitters. Okay, the second way is calcium channels open. It's a little more direct. May I erase the board?
Not yet.
Yeah, I don't know. I'll see through it over here.
Okay. So I'm going to erase it back here to the G protein. And so now what happens is the activated alpha subunit doesn't have to go through a whole bunch of other steps. It will bind to a calcium channel. And it will open calcium channels. Again, the receptor is not a calcium channel, but downstream, along floating in the membrane, is one. Now for hormones, this is not the last step, all right? For hormones, the calcium binds to another molecule, the calmodulin that you see there, and then it will go on to affect events inside the cell. But we're not going to focus on that as much. So this is not one we'll talk about a whole lot with neurotransmitters. This third event combines both of these. We activate a second messenger like cyclic AMP, and we open calcium channels. So this is not part of this event. Okay. This one is just calcium channels.
Protein channels,
Hold on
do
just
they
a
ever
sec.
just work on their own without the other one?
Say that again.
Is there ever an issue that's not hormonal? Do the G proteins and the calcium channels ever just work on their own
Without
without?
the calmodulin?
Yes.
Yeah, I mean, the calmodulin is probably there and doing other things, but the calcium coming in can also then activate function as an EPSP and cause voltage-gated channels to open downstream.
And what would it include?
It would be just like the sodium. It could be like a sonation. It could be added to the sodium coming in from a different neuron.
I mean, what would cause it just to do that?
Oh, I'm not saying it wouldn't do the calmodulin. I'm saying that would not be needed to open another channel. It could cause change enzyme activities inside the cell, inside the neuron itself,
Yeah.
and cause that to, it could change mitochondrial activity, and so on in the neurons, make additional enzymes inside the neuron.
But what would reason be for that?
If we have just an ionotropic, well, the cyclic AMP one could do that as well. So it could trigger the activity of other enzymes to synthesize other proteins that the neuron would need to make new microtubules, or to make new mitochondrial enzymes to increase mitochondrial activity, to recover, maybe for growth. So neuron is mostly involved in making some kind of a synapse, but we need additional enzymes to make neurotransmitters. We need additional microtubules, so we need to regrow dendrites and so on. It could cause it to proliferate the number of dendrites that are present. We need to make new proteins for that. So when we potentiate a neuron, we do learning. That's some of the things that occurs. New dendrites occur. So that needs new proteins. So there could be additional activities inside the cell as a result of the second messengers that are not opening channels.
So does that mostly happen when you're young then, when you're building those
No,
connections?
it can probably happen, well, it probably happens more often, but not only.
So this is a mechanism that happens all the time? I guess I'm trying to understand.
Yeah.
when-
So you're always learning.
Yeah.
Certainly there's more learning that goes on when you're younger, because the amount of new material that's out there that you haven't been exposed to exists. But you're always learning.
So that for us, the
Yeah.
thought process, why?
You would have additional activities.
So,
Yeah.
just the calcium channel.
You wouldn't say thank you if I just. I swear.
Okay, now the third thing, the phospholipase C, is kind of a combination of the first two.
Sorry.
So we're going to open calcium channels. We're also going to open membrane channels. The calcium channels that we open are more likely actually to be inside the cell. So this uses an enzyme inside the membrane that is different. Instead of being adenylate cyclase, this membrane is going to be phospholipase C.
I am.
Getting a better idea why I don't have you memorize all this stuff? And phospholipase C, when it's activated by the alpha G protein, takes another plasma membrane. So phospholipase C is abbreviated PLC. And this is actually a lipid. If you like long names, this has a really nice long name. Let's see, is that what I want?
Which is an enzyme.
Not quite yet. Now this is known as PIP2, phosphoinositol phosphate.
Say that one more time.
Phosphoinositol phosphate, PIP2.
Say that.
So,
We don't have to spell this one, right?
no ma'am. This is not the extra credit on the take
Oh,
home.
P what?
So when this is
P1, P2?
activated, it actually is split into two molecules. Remember I said there were two events that occur? So this one is split into a molecule known as inositol triphosphate, or IP3, and a molecule known as diacylglycerol, or DAG. IP3 opens calcium channels. Heard that before?
Yeah.
DAG activates protein kinases, which is what our cyclic AMP did.
So this is a combination of previous things.
It has a combined effect. It doesn't work exactly the same way, but it has a combined effect. Now, just to illustrate, we're going to get to this eventually, but here's acetylcholine. This is a pair sympathetic neurotransmitter. So here's the G protein. Here's acetylcholine. Here's the receptor. Here's the G protein. All right. The alpha component is excited and binds to phospholipase C. Phospholipase C takes phosphoinositol phosphate and splits it into diacylglycerol and inositol triphosphate, IP3. Calcium ions open. Diacylglycerol activates protein kinases, which bind to a potassium channel and open it. And actually activates the other proteins that bind. And so this opens potassium channels and becomes an inhibitory, because what we're doing, we're losing positive ions. Okay? So just to show you, this is a pathway that is used by acetylcholine to decrease your heart rate. This is what happens. You got scared by that CHP, okay? And your heart rate went up. Sympathetic neurons are going to be stopping releasing of norepinephrine. Then from your adrenal gland, it's going to be cleared away. But that's going to take a while. That would take even longer. So to bring your heart rate down, the parasympathetic, the vagus nerve, these are coming from the vagus nerve in your heart, it's going to release acetylcholine, and in your heart muscle, at the pacemaker, those cells are going to lose potassium ions, and your heart rate is going to decrease. Okay. One of the case studies that we look at next semester is a real true story about a woman back in the 90s, I believe, who in her early 30s, so she was a mountain climber. She had a goal to climb the highest mountains on every continent. She had climbed about 2, and she got sick, and it was a virus that attacked her heart. And at the age of like 32 or so, she needed a heart transplant. And so on her way to the hospital, I think it was at UCLA, she's flying over Disneyland, and she said she looked out and saw the Matterhorn and realized that she still had her goal of climbing these mountains. So she got her heart transplant. Now, what happens to your heart rate when you're climbing?
Goes up.
It goes up because of the release of norepinephrine. When they do a heart transplant, they don't reconnect the vagus nerve and the sympathetic nerves. So her only source of norepinephrine and epinephrine is from her adrenal gland. Well, that is a very slow process. The nerves can't just release norepinephrine and in milliseconds bind to their receptor. She had to wait, sometimes an hour and a half, for the norepinephrine levels from her adrenal gland to build up and reach her heart. And then her heart rate would start getting faster and she would get enough oxygen to continue with her climb. Okay? So now she reaches the mountain, and she continued to climb mountains. She's still climbing mountains. Now she's resting at the top of the mountain. She's going to be up there for a few hours before she comes back down. But she doesn't have the vagus nerve. She has no way of getting acetylcholine to her heart muscle. We have no hormone, no endocrine organs that release acetylcholine like we do that release norepinephrine. So it's going to take, normally our heart rate returns to a resting heart rate level in about 5 minutes if you're physically fit. It takes her over 2 hours. And she has to wait for the norepinephrine to be removed from the bloodstream. She has no nerve to muscle connection releasing acetylcholine, which is what normally brings our heart rate down and our recovery. So that 5 minutes doesn't work. 5 minutes doesn't work for her. Okay, but this is what's going on in the heart. It's using that phospholipase C pathway. All right. So those are the 2 basic types of receptors. And now we're going to look at neurotransmitters. So if you look in your lecture notes, because this is not something I'm going to talk about every single one. But if you look at your lecture notes, starting on page 34, I have classes of neurotransmitters. Single amino acids, okay, glutamate and glycine are single amino acids. Let me just kind of put these up on the board so you have them all in one place.
Glutamate.
Then we have modified amino acids. You guys think of an example of a modified amino acid?
Glutamate.
No, that's a single amino. These are single amino acids that have been modified. Who did Parkinson's disease?
It's the dope.
Yeah, dopamine is modified from what amino acid?
From the fan.
That's serotonin.
Tyrosine.
Tyrosine,
That one.
okay?
T.
Norepinephrine actually is also modified from tyrosine. Okay, so we'll look at that group. Even gases function as neurotransmitters. We have acetylcholine, which is its own little group here. We have larger protein. This is a small, and we have gases, such as nitric oxide. This is only in the last 5 years maybe that gases have been considered to be neurotransmitters. And then we have a class known as neuropeptides. Pardon? Yeah. Now, I've kind of allowed myself to mislead you by omission. When we were first talking about the neuron, when we talked about missile bodies, when we talked about anterograde transmission, I talked about the transmitter being synthesized in the cell body with the missile bodies, the ribosome or ER, and transported down the axon. Actually, that's not very common. That would be with the neuropeptides. Okay? But with these single amino acids and small molecules like acetylcholine, they're actually synthesized in the synaptic gluton. And axon terminals. So these would be synthesized in this region right here. They're often recycled. So we take acetylcholine, break it down into acetate and choline, and take it up and resynthesize it again, right there in the synaptic gluton. However, we still need that anterograde transport. What do you think we need it for? What proteins do we still need to be transporting, even if we're synthesizing? Huh? The mitochondria, but for the what's in the mitochondria that we need to be synthesizing? The enzymes. So even though we're recycling these, we still need the enzymes for those chemical steps. So when we put acetylcholine, acetate the choline back together, that's done by an enzyme. All right. When we break down, let's say we take up dopamine to get it out of the synaptic cleft, we break it down by an enzyme called monoamine oxidase. Where are we making those enzymes? Back in the cell body with the ribosomal ER. And that has to be transported down the axon via the anterograde transport. Okay? So we're still using that transport even if we're making this synthesis in the synaptic gluton. So I think we're ready for another short break. Okay? Just a short one, 10 minutes, and then we'll finish up once this comes on.
It's tired like
People ask me for them all the time.
the weather.
Like, do they, your hands are that sensitive? Do you know how many times I have to do that? Your hands are that sensitive that you can't hold on to an ice cup? They're not.
So single amino acids. The first one that we're going to look at two, glutamate and glycine. One is excitatory and one is inhibitory. Okay? So I do want you to know for the comprehensive, so underline this first statement here. It is the major excitatory neurotransmitter in the brain. What do we mean by excitatory? It creates
34.
EPSPs. And what does that mean? Is it going to cause the inside of the cell to become more positive or more negative?
Positive.
More positive. Okay? So it does so by opening up channels. And here we get into that. This is hard to understand because I don't know what the words stand for. So it has both ionotropic and metabotropic channels. It has several different types of ionotropic channels. All right? And here is the MDA channel and an AMPA and a kainate channel, as you can see here. These are all ionotropic, meaning they are the sodium channel. But this one is really cool, the NMDA channel. And that's because not only does it bind glutamate, but in experiments they found that they can test this with N-methyl D-aspartate. Aspartate is a type of amino acid that is methylated, and the D tells where it is. So this channel binds glutamate, all right? It is a calcium channel. But even after glutamate binds, it won't open. And the reason is because it's blocked by magnesium. This requires a higher membrane potential before it will open. So it's an ionotropic channel that is also voltage gated. So what happens? The AMPA channel is an ionotropic channel. So when glutamate binds to both of them, the AMPA channel will open and allow sodium into the cell. What's going to happen to the resting membrane potential? It's going to go more positive, and then it's going to remove the magnesium from the NMDA channel, and then finally calcium can come in through the NMDA channel. So it's this two-step process. The opening of the NMDA channel is delayed until enough sodium comes in. So at one time, glutamate binds to both of these. The AMPA channel will open immediately, allowing sodium ions to enter the cell. The NMDA channel will not open until enough sodium ions come into the channel to cause magnesium to leave. And then once magnesium leaves, calcium will come into the neuron. So the AMPA receptor, they're both receptors for glutamate, they're both ionotropic receptors. One opens for sodium, one opens for calcium, but both of them are going to increase the number of positive ions inside the cell. All right. NMDA receptors are thought to be involved in Alzheimer's, and they become defective in some cases of Alzheimer's disease, and seem to be the ones responsible also for some of the memory and some of the behavior problems with Alzheimer's. Also, glutamate is the key culprit in brain damage when someone has a stroke or someone has other causes of lack of oxygen to the brain. So for a long time it was thought when somebody had brain damage due to lack of oxygen, either from a stroke or being under the river for a while, because the cells died not having enough oxygen. What actually happens is cells that are dying to lack of oxygen release a lot of glutamate. What the glutamate does is then opens a lot of calcium channels, and that flood of calcium into the surrounding neurons basically destroys the mitochondria. And it's that effect that ends up with much of the brain damage that occurs. Is the subsequent effects of the glutamate damage rather than the original oxygen loss. And so current research is trying to minimize the glutamate damage. That is the secondary, and that of course is going to take a little bit longer, and it's something that surgeons and physicians, medical doctors have time to work on. So trying to block the glutamate from binding to the surrounding neurons and overexciting them can help to save them and minimize the subsequent damage. Okay, so that's glutamate. This is just a discussion of what's happening here with all the excess calcium and the free radicals that occur in the loss of the mitochondrial function. So that can lead to cell death. So then we have, those are the ionotropic, and we also have metabotropic, or metabotropic. This is using the cyclic AMP pathway that we talked about. Okay. So that can take a little bit longer to occur. So a lot of these neurotransmitters will have two different types of receptors, ionotropic and metabotropic. Some may be on the same neuron, some will be on one kind of neuron, and others will be on a different kind. We'll see that when we look at acetylcholine. Okay, glycine is another single amino acid. And it is one of two inhibitory neurotransmitters. How do you know it's inhibitory? What's our major negatively charged ion?
Chloride.
Chloride. So both glycine and gamma aminobutyric acid, I think the slides I put up online, it says GABA amino. It's supposed to say gamma amino. That's what GABA stands for, is gamma aminobutyric acid. It has two types. The ionotropic is directly for chloride. Okay? So this is going to result in an ESP or an EPSP or an IPSP?
IP.
An IPSP. You must have it now, Trish, if you could come up with the answer that fast, unless you were reading
Kind of,
it.
sort of.
Okay, so when chloride goes inside the cell, as you'll see in a few moments, it makes it less likely that that cell is going to fire, or if it's occurring, if we have a dendrite an axon at the synaptic gluton, it's going to prevent that from firing. Notice what else can bind to this site? Benzodiazepines, those are like Valium, is a benzodiazepine, barbiturates, anesthetics. Okay, so if we're releasing chloride and providing an IPSP, then a neuron that might fire and release in a pain pathway will be inhibited from firing. Okay, because of the increased amount of negative chloride ions present in the cell. So it's creating IPSPs. This also has a metabotropic pathway that results in the inhibition of cyclic AMP formation. So it's not going to work. Okay, that usually is because of the beta and the gamma activation rather than the alpha activation. The beta and the gamma activation. So notice here we're losing more potassium and we're not gaining calcium. So it has the opposite effect. All right. So in this case, it was an inhibitory because we added chloride ions in the ionotropic component. In the metabotropic, it's still inhibitory, not because we're adding chloride, but because we're losing potassium. So remember I said there were two ways to make the inside of the cell less positive. Either add negative ions, which is what we're doing here, or lose positive ions, which is what we're doing here. Okay. So it's more illustrating of that example than specifics that I'm asking you to memorize here. But those are two ways in which a cell can cause an IPSP. So here we have, not at the cell body, but this is the synaptic gluton. All right. this is, I moved this from another location, so this was on the PowerPoint, so I uploaded, but I moved it to this location. Here's the GABA being released. It is decreasing, it's creating IPSPs inside the synaptic bouton. It's making the membrane less likely to reach threshold. So now an action potential arrives, which normally opens calcium channels. But this action potential, because of the number of negative ions or the fewer number of potassium ions present, is not going to reach threshold for the voltage-gated calcium. So they won't open, so we don't release the neurotransmitter. All right? Now, then the next cell is less likely to reach its threshold and create an action potential. So we can have our axon terminals and our synaptic boutons directly affecting the synaptic bouton of a neuron or the cell body. So we've talked about this synapse always being synaptic bouton to cell body. It can be synaptic bouton to synaptic bouton and affect the release of those neurotransmitters. Okay? So does this make sense? If we make the inside of the cell more negative, when we have an action potential, it can't result in the release because the calcium channels are not reaching their minimum threshold. Okay. Now, occasionally we can have an axon to axon. So this what we've been talking about, just to take what we've been talking about typically is an axon to a dendrite, synaptic bouton to a dendrite. What I just showed you was this. So an axon to a synaptic bulb, synaptic bulb to synaptic bulb. You can also have an axon with this. So we have, this would be the axon. So if we release negative fluoride into here, what's going to happen to the transmission of our action potential? It's going to stop at midstream. Okay? So, again, this is what we've been referring to because that's a more common discussion of it, but we do have these other, or this one, we do have those other events. Okay. Monoamines now are the second class we talked about. So glycine and glutamate were single amino acids without modification that can work as a neurotransmitter. Monoamines are single amino acids that are modified, okay, in some way. And we have the catecholamines and indolamines. Essentially, it's just tyrosine and tryptophan are the two most common amino acids that are modified. And you've heard these words before. You've heard dopamine and serotonin before. All right. So those are the catecholamines. So here's dopamine. We take tyrosine and we modify it and we get dopamine. We modify dopamine and we get norepinephrine. We modify norepinephrine and we get epinephrine. So these are all just different forms of tyrosine. Okay? This has a hydroxyl group added to it. This one has a hydrogen taken off, a water molecule taken off, and so it's just various modifications of this component. So it's a modified, it's not tyrosine, but it's modified and they're very closely related to each other as far as the chemistry that's apparent. So dopamine works through a metabotropic effect. It can be here, the alpha protein is involved, and we end up activating adenylate cyclase through the alpha protein. Adenylate cyclase takes ATP and converts it to cyclic AMP, which activates protein kinases, all right, and we end up closing sodium channels. So when we talk about a G protein being excitatory or inhibitory, we're not talking about necessarily the ion changes. We're talking about whether we are activating the adenylyl cyclase or inactivating it. Okay? So in this case, the alpha protein is activated and we turn on adenylyl cyclase. Our end result is inhibition of the neuron. All right? But the G protein is excited. In the D2 receptor, okay, this is an inhibitory pathway. We turn off the adenylyl cyclase, we turn off the cyclic AMP, and now our sodium channel is open and the potassium channel is closed. So we end up inhibiting the G protein but exciting the cell. Again, you don't have to memorize this, but I just want to kind of introduce you to the concept of the complicated pathways that we can turn cells on and off. Some of them are backwards from others. What's making the difference in whether dopamine is excitatory or inhibitory? It's the postsynaptic neuron. Because dopamine is dopamine. So it has one effect on one cell, on one receptor, and a different effect on a different receptor based on the enzymes that are in that cell, in the postsynaptic neuron. All right? So that postsynaptic neuron is determining its own effect. So there's nothing different about the chemistry of dopamine. What's different about what that cell does with it. Okay, this stuff is just, again, for your information. Norepinephrine, we're going to look at a lot more detail on Friday, definitely Tuesday, when we talk about the sympathetic nervous system. But probably we should be getting to it on Friday. And it has both excitatory and inhibitory pathways. All of these are G protein pathways. So using phospholipase C, using cyclic AMP here. And again, we'll talk about those components when we get to the sympathetic nervous system. So we're going to talk about different receptors. Okay, so here it is, binding to its receptor. The G protein, here's the phospholipase C using the IP3 and the DAG. It looks very complicated, but it's that same IP3, PIP2 pathway. So there's a lot of commonalities in these different components. We'll come back. So here's norepinephrine binding to its receptor. In this case, it's using the adenylyl cyclase. All right, and again, sodium channels are opening. So as we'll see in the next couple of days, acetylcholine and norepinephrine can be excitatory or inhibitory depending on the cell it binds to. Okay? That's the key thing I want you to take away from this. Is what that excitatory or inhibitory effect depends on the neuron it's bound to. Okay, serotonin, change here. Serotonin is also known as 5-hydroxytryptamine. Remember, it's a modification of tryptophan. And so the reason I'm saying this is because the receptors are identified as 5-HT receptors. So I want you to recognize 5-HT as a synonym code for serotonin. For some reason, we don't talk about serotonin receptors. We call them 5-HT receptors. Okay? So if you're doing research for the take-home exam, you're going to see 5-HT rather than serotonin. Or you might want to look up 5-HT. Okay? And we're going to come back in a few minutes and talk about what we do, how we get rid of, or how we stop the activity of these different components. So here's a 5-HT receptor. We block it. These are done by reuptake. We're going to come back to these in just a sec, just a moment, like in 3 minutes, and talk about what happens after the neurotransmitter binds to its receptor. There are 3 major ways that we get rid of it. So here's the ionotropic one. Just by looking at that, after today, you should recognize that as ionotropic. Sam, you're shaking your head.
It's not about this.
Holding, okay. Because the fact that it's an ion channel, all right? If we look at, so when, here it is, it's saying 5-HT instead of serotonin, but it's the same thing. So when it blocks to its site, it's going to open and become excitatory. Now, it also has a metabotropic. So here it's using the cyclic AMP component and opening potassium channels. If we open potassium channels, is it excitatory or inhibitory?
This one is inhibitory.
Inhibitory, because we're losing positive ions. Okay, so those basic things that I'm repeating over and over, I want you to understand that concept. When we lose positive or gain negative, it's going to change and decrease the possibility that we reach a threshold. Okay, so the pathways, so here's serotonin. Okay, this is what would be an excitatory part of serotonin. So it's going to increase the sodium inside the cell. So if we use the ionotropic pathway, and we increase the sodium inside the cell, now when the action potential arises, it's more likely to open calcium channels, and even more calcium channels will open than normal, and even more of the neurotransmitter will be released. So there are certain drugs for depression that increase serotonin levels, and this is the result. Downstream, there's an increase of other neurons that are excited. So in 1 minute, we'll look at how we modify, how do we increase or decrease the level of serotonin. I want to finish up with acetylcholine. So the acetylcholine is our neurotransmitter for the parasympathetic nervous system. Acetylcholine binds to a receptor. Again, it has two different types. Acetylcholine has ionotropic receptors. Okay, we're going to talk about this on Thursday, so we're going to come back and look at this slide on Thursday. So it's going to open an ion channel. These are always excitatory. Okay, so for acetylcholine, ionotropic receptors are always excitatory. However, the metabotropic receptors may or may not be excitatory. Some can be excitatory, some can be inhibitory. The M, we'll talk about this on Thursday. The two different types, another name for the ionotropic is nicotinic. Don't bother writing that down now because we'll talk about it on Thursday. Another name for the metabotropic is muscarinic. What that means is, when they were researching this, the ionotropic would preferentially bind nicotine. When they were researching the metabotropic, they preferentially bind a chemical from a certain type of mushroom, and so they call those muscarinic. There's several classes of muscarinic receptors, and some are excitatory and some are inhibitory. If they are inhibitory, here's an inhibitory one. Notice which kind of channel it's opening? Potassium. So this would be this is the slide I showed you when we were talking about the heart transplant. So this would be how it decreases the heart rate. It causes those cells to lose potassium ions, so it takes them a longer time before a new heart rate, a new impulse is stimulated to cause the heart to contract. Okay? So again, the general concept of the fact that one neurotransmitter can be excitatory and inhibitory depends on what kind of channel in that particular cell it opens. And that depends on the ions that are in the cell. Okay? That depends on the pathway that that particular cell has. Acetylcholine is excitatory to the urinary bladder. So if someone that, every time they talk about people that have problems with their bladder, going to the bathroom too often, having bladder spasms, they seem to show guys riding their bicycles in the rain or kayaking in the rain. They're always in the rain or the water or something, okay? So if acetylcholine stimulates, that would be where it's doing sodium ions. If it stimulates the bladder, we want to stop acetylcholine from working, okay? Sometimes people have trouble going to the bathroom. So after surgery, it may take them 3 or 4 days before their bladder responds, and they have to be catheterized because the muscles won't relax and allow the urine to leave the bladder. So how do we control the level of the neurotransmitters? And this is where we're going to finish. How do we control the level of the neurotransmitters in that synaptic cleft? There are 3 different ways that can be modified. So the first mechanism is illustrated with dopamine. Dopamine and norepinephrine and serotonin were all monoamines, right? Tryptophan and tyrosine modified amino acids. So, what happens in many of these is after the neurotransmitter is released and it binds to its receptor, there are other neurotransmitters in the synaptic cleft. We don't want them to stay there forever. Otherwise, there's no point in activating this neuron. We're just going to turn it on, and it's like walking out the bathroom and leaving your bathtub faucet running forever. Okay, it's just going to deplete the cell of the neurotransmitters, and we're going to lose our reason for having a signal. So what happens is the cell has transporters, and it takes the neurotransmitter back up into the presynaptic neuron. So this neurotransmitter is going to be returned to the presynaptic neuron. It's called reuptake. Within the neuron, it interacts with an enzyme known as monoamine, from our monoamines, oxidase, thus MAO. All right? So one mechanism for breaking down or modifying, affecting the fate of our neurotransmitters is reuptake. And recycling, breaking down and recycling by the presynaptic neuron. Now a sub-step to this is that sometimes our astrocytes will take the neurotransmitter, break it down, and then send those products back to the presynaptic neuron. This is what happens with glutamate. It can be broken down and recycled and sent back. Well, it's actually converted to glutamine, and then sent back to the neuron and reconverted to glutamate. Okay? So this may be done aided by astrocytes. Now, there are two major ways in which we can affect this. What if so I don't have enough dopamine, all right? I currently take a drug that works as a dopamine agonist. It acts like dopamine and binds to dopamine receptors. My migraine medication is an MAOI. It's a monoamine oxidase inhibitor. What that does is it prevents monoamine oxidase from working and increases the buildup of dopamine or serotonin in my synaptic cleft. which is why it's contraindicated that I take both of those drugs at the same time. So my physician, I used to take a codeine-bound drug, Furanyl plus codeine. And about 2 years ago, my family physician, when I called to renew it, my prescription would be for about 25 pills, and they last me anywhere from 3 to 8 months. Okay, they called me in and said, we're not renewing your codeine prescription anymore because this office is no longer going to dispense codeine. It's an addictive drug. And I said, well, okay, I understand that, but I don't take it every day. It can last me up to 8 months. And I can understand if I took it 2 pills a day that I would get, I could get addicted to it. Well, it doesn't matter. Nobody prescribes codeine in Europe. I was like, well, okay. But I understand, so what are we going to do instead? So they gave me a couple of samples of a migraine drug called Relpax. So the next time I had a migraine, I used it, it worked fine. So 2 months later, when I had another one, I called for a prescription, went to fill it. The pharmacist said, it's a new drug, so he said, okay, let me go over this with you. And he looked down and he goes, you need to give that back to me. You can't have that. I was like, but I just paid for it. He goes, I don't care. You can't have that. Give it back to me. He said, we'll give you a refill. I said, what are you talking about? He said, you're already on bromocriptine or Parladel, which is the dopamine agonist. He said, this is a MAOI, and it can cause serotonin crisis because the two amplify each other, and you have too much serotonin, and your blood pressure goes high, and it's fatal. I was like, oh, okay. So I went back to see my physician, and he said, I don't believe you. And I said, look it up in the physician desk reference, the PDR, and it's the very first drug listed as being contraindicated for each other. And so he said, well, okay, let me talk to a friend of mine who's a neurologist, and I'll get back to you. And he called me and said, well, the neurologist said it's okay. So now every time I get it, I have to sign my name that I understand the risks. And the bromocriptine comes from my OBGYN, so every time I get that refilled, they call me and say, do you know that you're also? And I'm like, yeah, I do. And I haven't had any problems, but I am aware of what the symptoms are to look for. I take the bromocriptine every day, twice a day, and the Relpax about every 2 or 3 months. So, an MAO inhibitor, MAO inhibitor is one way of blocking that. Okay, another way is working on the transporter. So some drugs, dopamine has been implicated in ADHD, attention deficit hyperactivity disorder. And so some of the drugs affect this transport, or even the gene for the transport protein. Either block it or change its function. All right, so a lot of these drugs work through this. There's another type of a drug specifically for serotonin known as SSRI. Obviously, one of the S's stands for serotonin. The other S stands for selective. So it's selective serotonin reuptake inhibitor. So again, it blocks the transport here. That's going to leave more serotonin. A lot of mood elevators work that way. Okay, so that's one mechanism by which it works. The second mechanism, we're going to look at acetylcholine. Oops, can I erase the picture? So now let's look at acetylcholine. And acetylcholine is actually broken down by an enzyme produced by the postsynaptic neuron. So if this is acetylcholine, there is an enzyme present in the membrane of the postsynaptic neuron identified as acetylcholinesterase. And it's going to bind acetylcholine and break it down. Because when our neurotransmitter binds to its receptor, this is not an irreversible reaction. That would be terrible. okay? Because then we can never get rid of it. So it's a reversible reaction. So the more we remove acetylcholine or any neurotransmitter from the synaptic cleft space, when this comes off, eventually, which it will, there is not another one to take its place. So by reuptake or by breaking it down by acetylcholinesterase, we decrease the possibility that any given time a neurotransmitter will bind there. So let's say I'm playing musical chairs, all right? And I have 10 people playing musical chairs, and I have 5 chairs. So when the music stops, all the chairs are going to be occupied, right? Which is what happens if we have a lot of neurotransmitters in our space here. At any given time, when this neurotransmitter comes off, there will be another one right there to take its place. So it will be as if it never came off. All right. Now let's say that we have 15 chairs, 20 chairs, and only one person. The music stops. Only one of those chairs can be occupied. The other 24 won't have anybody sitting in them. So as we decrease the number of neurotransmitters, we increase the likelihood that this will not have a neurotransmitter binding to it, and that channel will not be activated. So when we look at the parasympathetic nervous system, let's say that somebody is fresh out of surgery. Maybe now it's 3 days and they still can't go to the bathroom. We want to increase the amount of acetylcholine that they have. We can do that by giving them a cholinergic drug that mimics acetylcholine, or we can do it by blocking acetylcholinesterase, an anti-cholinesterase drug, which will keep this from working and increase the amount of neurotransmitter in the space. So the second mechanism by which we affect what happens to neurotransmitters is using enzymes in the postsynaptic membrane. And that's what I illustrated here with acetylcholine. The third one is really simple. Just diffusion. A whole bunch of neurotransmitters in here. Not all of them are going to stay, and some will eventually just move out and get removed by microglial cells. Okay. Except for acetylcholine and norepinephrine, that's all we're going to talk about neurotransmitters. We'll come back and revisit those when we talk about sympathetic and parasympathetic, because they have. But we're not going to talk anything new about this. We're just going to talk about what they do, not anything new about their transmitters or the receptors. All right. So the take-home exam is almost ready. I'll post it tomorrow evening. It's not our lab exam is 2 weeks from today. That's over all the stuff we've already had on the brain, and the new material on the eye and the ear and the autonomic nervous system. But about 3/4 of it will be old material, 2/3 to 3/4, but closer to 3/4. Okay, will be older material. We'll have a lab Thursday and Friday on autonomic, and then we do the eye and the ear next week. Your quiz on Thursday is going to be a review of the old. So take another short break. We'll look at some of those slides, okay? And I'll tell you which one will be on Thursday's quiz. It will be the model with the thalamus and the brainstem. So it'll be limited to those structures. Thalamus, fornix, third ventricle, cerebral aqueduct, medulla pyramid, superior colliculus, cranial nerves. So we'll go over some stuff already. So take a break, go outside,
Just breathe.
slap your arms around.
Didn't hear you.
Drink coffee? No. Hot chocolate?
Hot chocolate, cold chocolate, warm chocolate.
Hershey's Kisses?
Хорошо.
Halloween is over, I'm done eating candy corn.
They have the little.
This is the only time I eat candy corn. They had it in the off-site shop. I had about 3 pieces a day.
And bland.
And I'm done now. Candy corn at home is all gone.
Right?
Making my way back to my area.
All right. So, the structure indicated by the arrow,
Neurolima.
neurolima, very good.
And what is neurolima?
It's a layer.
Isn't it the two things?
It is the outer plasma membrane and underlying cytoplasm of the blood cell. Okay?
Is this on your PowerPoint online?
No. There's a ton of practice. I mean, yeah, the information's online on the practice page.
Yeah, well, I want.
Okay, identify the structure.
Is it
Myelin sheet.
the myelin sheet?
Okay.
We're holding feet.
That's
We didn't really talk about this in endoneurium. So we'll pick this up when we do muscle and we talk about the peripheral nerve. This is the peripheral nerve.
The last one, myelin sheet.
What's the cell? The neurolima is not the answer. Schwann
Schwann
cell.
cell.
Okay.
Lateral sulcus.
Lateral sulcus.
Central
We
is in the
make
middle.
this.
The
No.
the
We're not too far forward to see the insula yet. So it would be
Temporal lobe,
the
right?
temporal lobe. Yeah, actually we're too far backwards. The
that's
answer's
central.
not there, but what is it?
Oh, ladder.
It's not, the answer's not there, but what would that groove be? Yeah, the longitudinal fissure.
Iris.
So look where we are, we're in the third ventricle. We can see the thalamus, so yes, we would be at the level of the post,
Post-central gyrus.
yes.
Cingulate gyrus.
Cingulate, remember you're used to seeing the cingulate from a sagittal view, but it is just above the corpus callosum. Mammillary bodies, good.
Mammillary. Okay. Mammillary bodies. Thalamus? No, globus.
Thalamus,
That's thalamus. Globus pallidus. Those are the globus pallidus.
That's the same.
Yeah, Tamen or
Oh,
Tumen. Globus pallidus has disappeared,
oh.
so we're towards the posterior part of the third ventricle.
Is that the form?
Fornix,
Fornix.
okay. Same thing.
Caudate.
Caudate, Be the body of the caudate at that point.
Substantia.
Substantia nigra. Okay.
With the dope.
Yes, that would be the head of the caudate.
Tail.
No,
Tail. It's not the tail.
or what part of the ventricular system are we seeing?
Is that the anterior horn?
We're in the anterior, no, we're in the inferior horn, we're in the temporal lobe. Anterior horn would be up here. So what forms the floor of the inferior horn? Hippocampus.
Oh.
So follow this structure, this is the hippocampus. See how this comes up here? So here's the head of the caudate, the tail has been removed so we can see the hippocampus. Because the tail of the caudate forms the root. So that's been cut out so that we can see the hippocampus. So what would that be then? Cortex, leading over the thalamus from the hippocampus. So here's the hippocampus, and then leading from that over the thalamus down to the mammary bodies is the fornix. So that place right there is where we saw this particular, that's what this is right here. So that's fornix shown at this place right here. Okay? What's that? Thalamus. Excellent. Very good.
Corpus callosum.
Corpus callosum. As is it, this is as well. Okay, so this is not a neuron model that we have, it's one we have at SAC state. So that would be the synaptic gluton of another neuron. It doesn't belong to this one. So I just gave my lab exam yesterday at SAC state on this. Actually, I had that very thing tagged here. And my students, some of them that missed it, were calling this a dendrite. But the dendrite would be for this neuron here, right here. And this is the axon terminal and synaptic gluton of another neuron, the presynaptic neuron, synapsing with it. Straight sinus.
The cut.
Inferior sagittal. Now this would be the inferior horn of the lateral ventricle. So at the bottom of that is where the hippocampus is, and in the top of it, the tail of the caudate. Cerebral aqueduct. So this will be the model that you're quizzes on Thursday. Middle, cerebellar peduncle. Inferior is not shown, but I usually show it as a dot. I mean, it's not a separate structure. I usually show it as the lower portion, right, just a little bit down here at the bottom.
In the middle.
Okay. So that right there, like that.
Okay.
That's the inferior.
So which part of the corpus quadriculum? The inferior colliculus. Okay.
Is what? This is no.
Yes, it is. Fornix. Huh?
Like that.
It is the fornix. Remember where the fornix comes? If we look at this picture back here again,
Oh god.
get to it. The fornix comes together over the thalamus. So we're seeing the fornix right here and right here in that particular model.
Seizure.
Okay, so fornix. On the side here, it's pointing to the side. Cerebri or cerebral peduncle. Dorsal view. Okay, because we can't see the peduncles quite as well. But on the quiz on Thursday, it would also show the ventral view. From the ventral view, we see the cerebral peduncles. We see mammillary bodies. We see the basal pond, the medullary pyramids. We see the stalk of the pituitary, optic tract. All right, a lot of those cranial nerves. So looking at the spinal cord, what are we seeing here? Not ramus, dorsal root. The dorsal root is directly associated with the posterior right horn, lateral. And we'll see this, talk about this on Friday because this is the location of the motor neurons for the sympathetic nervous system. Corpus callosum. All right.
Septum pellucidum.
Nope. Fornix is here. Septum pellucidum. There's your fornix. Thalamus still.
And then the
Hypothalamus.
dura mater.
Dura mater. So what you cut off of the sheep brain. So what would we be seeing?
That's essential.
Actually, it's not shown. It should be the transverse right here. So this would be straight, transverse on either side.
Cardiac
Huh?
horse's tail.
Filling.
Okay, good. You guys came back easier than you thought? All right.
So, know which one is the one that
The brain stem will be for Thursday. All right, so on the Physio X, has anybody not tried it? Okay, good. Everybody's tried it. Problems? How many of you have done the 8.0? When you open up my AMT, 8.0 is the one that opens up. You can switch to the 9.1, or if you have your books, you can use that one. All right. Let me just show a couple of things that are not present on the 9.0 that are present on the 8.0. I actually have it here. I don't believe this experiment is on the 9.0, but the concept is one that I want you to talk about. And it might be, I'm not sure.
It's right there.
So what we have here, the first experiment on the 9.0 starts off with a single neuron. And this one shows, there's a difference here. This one is showing the sciatic nerve. All right, so as we try to find the resting with the threshold stimulus, I'm increasing the voltage. So when I give a single stimulus, you can see that nothing happens. As I continue to increase the stimulus,
Bam.
now I get one. Now let's drop it down.
Oh, not doing it. Tired.
Let's see what my minimum is here. Nope.
Bam.
Okay. So now I talked about the all or none response. What does an all or none response mean?
Either you have enough or you don't.
Okay, if I get to threshold, I open up my voltage-gated channels, and I reach the action potential. I'm not going to get a stronger action potential. I'm not going to get more positive ions into the cell if I use a stronger stimulus. So now let me increase my stimulus, and well, what happens? My action potential is higher, and my hyperpolarization is lower. But I just said that the all or none response means that I can't do that. So how does this work? The key thing was in the very first statement I made when I brought this up, and I described, what did I say this was?
Sciatic nerve.
Sorry.
Sciatic nerve. How many neurons are in a sciatic nerve? Thousands, hundreds of thousands. Each of them, as an individual neuron, follows the all or none principle. So at 3.0, I match the minimum threshold for all those neurons that had that as their threshold. But there were a whole bunch of other neurons that had a higher threshold stimulus required. So I had to turn up the stimulus to reach their threshold, and they had slightly higher action potentials as well. But that statement, all or none, holds true for one cell. Okay? But this is more than one cell. So that's why it took me a greater degree to show that.
How high does it go?
I don't know how high this number goes. Here goes up. Let's see.
See what happens.
Let's see how high we get it.
Well,
Yeah.
it just shows it slightly more different.
Yeah. Badass.
All right, now,
We got a plug.
you'll notice over on the left are some drugs. One is lidocaine. So let me increase this to my minimum. Make sure I get it. Okay, that works. Now, if I bring lidocaine over here and drip it on my nerve and then stimulate it, I don't get an action potential. Okay? So on your lab write-up, some of them are lidocaine, one's curare, one's tetrodotoxin, which is the pufferfish, that Japanese delicacy. So I ask you to give me the mechanism of action. It's nothing to get full credit. I don't want that it inhibits or excites. I want you to tell me it is an agonist for the sodium channel, or it blocks the sodium channel, or something to do with what channel does it affect, does it open or close it, okay, for the full credit. Now, so lidocaine actually works by blocking sodium channels, so it's going to inhibit voltage-gated sodium channels, so it's going to inhibit the formation of an action potential, kind of like adding a whole bunch of calcium. We talked about that earlier. So now let me flush this. And let's put curare. Curare results in paralysis. It's used sometimes in surgery, but we have more sophisticated drugs now. to stop somebody's respiratory muscles so that the anesthesiologist can use the machine to breathe for them, stops them from gagging as well. So let's put Ferrari on here. Grip, 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 grip. And now let's stimulate. We get a stimulant. So what does that mean?
That it doesn't block?
It's a trick question.
It's too high a voltage.
Curare blocks acetylcholine. It is an antagonist. It binds to the receptor for acetylcholine, which we know is a neurotransmitter, right? So why didn't it work?
Because we're saying neural.
Because the curare, the acetylcholine receptor, it blocks is on the muscle cell. So the nerve still works. It still releases acetylcholine, but that acetylcholine is not allowed to bind on the muscle cell, so the muscle does not react. Now we're stimulating the neuron, so we still see the neuron activated, but the muscle cannot respond. So muscle shares receptors for acetylcholine. And we haven't talked about muscle yet, okay? So this activity is assuming that you've already had the muscle chapter, but acetylcholine binds to muscle, not just to nerves. And so this is curare works specifically on muscle acetylcholine receptors, not nerve acetylcholine.
So you could feel pain, but you couldn't move.
Correct.
Awesome.
That's
So if you came to during the surgery, you
Yeah.
would not be able to tell them that the anesthetic has washed off. There's actually a murder case in Texas where a nurse was married to a physician. I believe, obviously, in most cases, she was having an affair. He was very rich and very old. Anyway, she wanted him to die. And he was spending money on his daughter's marriage and stuff that she didn't want him to spend money on because then she couldn't spend it. So she brought home a similar drug. It wasn't, I don't know if it was scopolamine, but it was similar to curare, and injected it with him, into him. He was still conscious. It took him a long time to die. It was a respiratory, so it was paralyzing his respiratory muscles, but she didn't give him a fatal dose right away. But he wasn't able to move or blink his eyes or do anything but just take in all the sensory part, knowing he was going to die and not do anything about it, which was part of the extenuating circumstances that the prosecution brought about in her sentencing. But there are other drugs that do have a similar effect. Okay, so those are a couple things I wanted to bring out on the 8.0 that isn't shown on the 9.0 or the 9.1. Okay, does anybody have trouble accessing these? Okay. So this will be due on Thursday. Give you a little bit more time to work on it, since we've covered over some of this material today and reviewed it that you might not have had available when we were working on it. All right, last thing, I have your exams.
I don't want it.
So did you get the notice that the grades have been posted before the email?
Yes.
I see, there, right. Okay, which one do I hit first? Okay, Karina, Bartosz? On the grades, there were 20 points available from the cranial nerves, which I thought was a little high for the amount of time we spent on them, although there was a lot of information. So I only counted 15. Originally, there were 8 points on extra credit, which means there were 105 points, and I divided by 92. All right. So there were 97 points possible from the exam, plus 8 points from the extra credit, and I dropped it to 92, eliminating 5 of the cranial nerves, but I just